So today, I'll be talking about generative models that have been developed in natural language processing and related fields for use on sequence data in preparation for Debbie's talk on how these can be used in biology. So all of us have now access to these powerful natural language models in our daily lives. There's a good example, which is digital assistants like Siri, which convert speech to text, then generate responses to queries, and convert those responses to speech. All of these require very sophisticated models of language, and the other examples include apps that are able to translate speech or text between languages, and they're also able to generate text from speech and recite translations back to you. All of these models have seen a massive increase in performance in the past few years. And here I have an example, which is a task of machine translation between English and German, which is measured with a blue score, which is a standard in the field. And since 2014, there have been a series of models that have made rapid progress towards the performance of the best human translators, which are at 40 blue, and now the current state-of-the-art has reached 35 blue. So there have been a series of advancements in neural network architectures and efficient training schemes that allow us to reach this performance, and I'll be outlining the developments in these models. So to go back a little bit in models of language, if we zoom out, this is a very general, non-comprehensive outline of popular approaches to language modeling, and when they first kind of became popularly used for the task, starting with n-gram models, also called K-mers in biology, followed by hidden Markov models, first used for speech recognition in the 70s and 80s. And then there's different neural network architectures, including a set of 3 architectures that I'll be discussing later that appeared in widespread use in the 2010s. So these models, all of them except for the hidden Markov models, are examples of autoregressive generative models. They have that in common. And so I'm going to be discussing, in turn, what are generative models and then autoregressive generative models. And finally, I'm going to talk about how to sample from these models, and then a little bit about how they might be applied to biology. So what are generative models? In these models, we want to model the joint likelihood of a set of variables, x1 through xl, and these set of variables could come from anything from text to audio or images, music, or some biological data. And we want to model the joint probability of all of these x's to obtain the probability that that set was generated by some underlying unknown process that we can't observe. And it turns out that these are great models for a lot of things, including evolutionary processes, as Debbie will be talking about later. But I'm going to focus on how these can be applied to language. In general, they can be used to generate new sets of x's that are synthetic and that match the underlying processes that generated those data. They can be used to score the probability of observations as having come from those processes, and we can use these to have perhaps some insight into the processes that have generated the data. So in language, an example of a generative task is the Shannon game, which originated in the late 40s and early 50s from Claude Shannon. And it's a simple task where we're asked that given the last n words in a sentence or a piece of language, can we predict the next word? And this is kind of related to the entropy of language. How predictable is language? So for a given sentence, like I took my dog for a blank, we would try to fill in that blank, and like banana might not make sense unless the dog is in some funny pose. But perhaps like walk, we'd intuitively fill in the blank with that. Or perhaps swim might be a second option that's a bit less likely. But we also know that there are context dependencies within this piece of language. So if we swapped dog for something else, then the options would change. Or maybe if we swapped took for something else, like if we swapped that to walk, I walked my dog for a blank, then maybe we would say that this should be a span of time or something. So I walked my dog for an hour. So there are rich context dependencies within even this small example of a piece of language. Now, how do we model this probabilistically? A natural fit for the Shannon game is an autoregressive generative model. So this is where we take a sequence of variables and we factorize the joint likelihood of the variables, p of x, into conditional probabilities of each x, the product of conditional probabilities, given the previous variables x. So to do this, we have to choose an ordering for the x's. And this is pretty easy for a sentence or a piece of language or some other time series data where there is a natural ordering. But autoregressive models can also be applied to generate pieces of data that don't have such a natural ordering. One of these is spatial, where there's relative positions that are close to each other, but there's no one-dimensional ordering unless you use special masks, as in these pixel by pixel generative models. But there's also a random ordering if there is truly no natural ordering to the data. So this might be used in, say, if you're generating a model of gene expression across a number of cells. Perhaps like you don't want to consider that any gene is necessarily closer than another. And so in that case, we could generate these in a random order. So autoregressive generative models, the directed graph that I've shown here is actually a fully connected graph. So in theory, we could model even the most complicated of p of x's. And yeah, so once we've chosen an order, we do the factorization. So how does this compare to other generative models? In particular, undirected graphical models or directed graphical models that have hidden unobserved states. Well, the great thing about these is that we have an exact probability of the data without any hidden states, which means that we don't have to use approximation strategies to train the model. This means it's very easy to train by maximizing the likelihood of each of the conditional probabilities xi. It's also easy to sample from this model because we can sample each x in order, though that is slow because we have to do things sequentially. Kind of the con of this approach is that there's no natural latent variable within the probabilistic graph itself. Though there are ways of expressing latent representations of the data within a certain parameterization. It's not explicit within that graph. And so, yeah, these choices, of course, depend on domains and applications, but in natural language processing, these are a very common choice. So the simplest autoregressive language models, the simplest example is where we only consider the n a very finite, a very small set of preceding x's within an ordering. So in the top row, I have what are called bigrams, where each x only depends on the preceding x. But we can kind of adapt this to an arbitrary n number of preceding states. So bigrams and trigrams were first used by Claude Shannon in the 50s. But when we extend these, we can have kind of a more complicated model of a given number of n. So now, if we wanted to approximate the autoregressive likelihood conditioned on everything, we could just expand the n to some very large number that could cover any length of sequence that we'd want to look at. But the problem with that approach is that the number of weights, or the number of parameters in our model, then scales linearly with that n, which makes that kind of intractable for very long sequences. Nevertheless, these models are capable of doing some interesting things with modeling language. So even though it is a very short context, it can be used to generate somewhat plausible text. So with a context of 4 characters, and using those 4 characters to predict the next one, you can fit a model that looks vaguely like English, in that it has words and and phrases somewhat. This is a model that was trained on Shakespearean works and then used to generate next character predictions. And as you increase the context from 4 to 10 characters, things look a little better. But still, on the level of sentences, even, there's not necessarily that much coherence to anything that's being generated. So how can we improve these models by increasing the context further? There are some previous approaches, kind of augmenting n-gram models with a cache of the previous words. This is based on the observation that rare words become more likely to reoccur if they've been seen earlier in the model. So that's one sort of representation of prior context before the n that we'd be looking at. It's not the most informative context, but it does improve performance. The other way is to have a very large network where every x depends on all the preceding ones and has a weights for all of the preceding x's. But this means that the number of parameters scales with the length of the input squared. So that's also fairly intractable for long sequences. So is there a better way to incorporate all the context in an informative way, but for the model, but that is also parameter efficient? What became the most popular approach to this was the recurrent neural network. So this is where it can incorporate a theoretically unlimited context by propagating a hidden state h all the way from the beginning of the text to the current position where you want to predict a character. So hi is a transform of the current input plus a transform of the previous hidden state as a nonlinear transformation. So this allows us to propagate information all the way from the beginning. These are called Elman recurrent neural networks, and they were used in large language models starting in the 2010s. Even though they were used in smaller scales before then. And there's some pros to this, which is you can have this unlimited context, and it's parameter efficient because the number of parameters doesn't depend on the length of the sentence. However, there's the con that the hidden state is an information bottleneck for all of the dependence on previous states, as the size of the hidden state doesn't increase with larger sentences. That can be a pro as well if you want a fixed size representation for a sentence, but it is a limitation in that it makes this model hard to train. They're like, if you want to compute dependencies between 2 positions, it takes you have to do matrix multiplications on the order of that distance to relate those 2 positions. So yeah.
How how many dimensions
is typical? So I think it can be on the order of hundreds to thousands. The hidden state H in language networks where this is commonly used. Right. Yeah. So if you want to relate two positions at a long distance, you have to do the sequential matrix multiplications, which also means that the gradients that are used to train the network can either go to 0 or explode, which is another complicating factor for training these networks. To address this vanishing gradient issue, there's a development of these networks called the long short-term memory, where there's a separate cell state, C, capital C, that goes along the top of this network diagram. And the model is much more careful in how it modifies that state over time. So it uses like a forget gate at every step to decide which information to mask, and then an input gate and this update wait to decide what information to add to that propagated cell state. So this is this was from Hackreiter and Schmidhuber in 1997, and it was used in large language models starting in 2013. And I guess the advantage of this then is because it's more careful with these long distance dependencies, it can be trained over longer context. It can include or incorporate longer contexts. But I guess it retains the con then that training still has to be sequential over the data. This makes it hard to parallelize on a large-scale compute. Nevertheless, these models, if we revisit that translation performance graph that I showed earlier, these are sufficient to get us to the 2016 state of the art, which was Google's neural machine translation algorithm. And it was powerful enough to allow Google to switch from this 500,000 line model of phrase-based translation to the single 500 line, much simpler model that can exceed that performance. So, and also like they can generate better Shakespeare. So if we revisit that Shakespeare generation, now we have more complete sentences, though they don't always make sense. Yeah. So one thing that LSTMs can do that n-gram models cannot is they can capture these long distance dependencies. So, for example, like even with no prior knowledge of code structure, if you train an LSTM on computer code, then they can learn that, say, open braces should be closed, even at long distances. Yeah, they are able to capture these, but their ability to capture long distance dependencies depends on how complex the structure is of those dependencies. So LSTMs are powerful modern models, but they're slow to train. And so we want to figure out how to improve this. And one way to do this is to use convolutional neural networks. And here we're going to kind of revisit that n-gram model, because a convolutional neural network where we pass a convolutional filter U over the input and stack multiple layers of convolutions allows the model to, in parallel, calculate the weights or the hidden states for every input at the same time. However, like the n-gram models, there's a fixed context. Right. So these were first used for pixel by pixel image generation. And yeah, it's parallelizable. But the con is that there's a finite receptive field, and if you want to increase that receptive field, you need to add on the order of the distance, add layers on the order of the distance between inputs, which means that the parameters, or the number of parameters, is on the order of the context that you want to capture. So how can we expand the context in a more parameter-efficient way? We can do this using dilations. So as we add layers, we also dilate the convolutional kernel, or the space over which that kernel is applied. So there's a multiplying factor within that dilation within that convolution operation. So this means that we can have this exponential context. These were first applied to model audio and translate between languages with WaveNet and ByteNet, respectively. And the pro is that now we can connect two dependencies with log of the distance between those two positions, as compared to a linear relationship between the distance. But again, I guess this model has the limitation that the receptive field is finite, even though you can expand it fairly easily by adding a few more layers, since the receptive field is exponential with the number of layers. So one application where these dilated convolutional networks is particularly well suited is when in generating raw audio. So audio has this interesting property where it has very fine structure, but also as you go into the scales of thousands of or hundreds of thousands of samples within audio, there are also very long-term dependencies. So if we wanted to model this with a RNN, or with a recurrent neural network, this would be a lot of steps to go through for every training example. So it would be slow to train, and there's a lot of steps where the model could lose long-distance information. So Google, yeah.
Except the field is small, then why does it work better if the long-term dependencies are like more important in this task?
Yeah, so with the dilated conv, let's see, going back a little. Okay. So the question was, like, if the receptive field is limited, how do we capture long-distance dependencies? Is that okay? So yes, the receptive field is finite, but it's also easy to expand. So if you have a sufficiently deep network with enough with a long enough dilation factor, then you can capture those long-distance dependencies as long as you wish to specify. So you have to know in advance how long you want to look, but you can specify a model that can capture those. Okay. Yeah, so Google used this model to improve the performance of their text-to-speech algorithm. So this is a comparison where humans were asked to rate generated audio from a few different algorithms of Google's. So there's the concatenative and parametric algorithms, which were the previous state of arts that use recurrent neural networks, or HMMs, to capture long-distance structure or large-scale structure, and then more simple models to generate the raw audio itself. But when they used the single model to generate every sample in audio, and there are tens of thousands of samples per second, they were able to improve performance, though it doesn't quite reach human speech. And also, these are human ratings, so that's kind of the only way to evaluate these data. So, dilated convolutional networks can aggregate long-distance information very well. but there is still a finite receptive field, and it takes a number of layers to correlate to very long distance positions. So is there an even more direct way of relating to positions? This is where self-attention networks come into play. This is where you can directly, in one step, synthesize all of this information. So how does this work? First, we have to compute a transformation, a linear transformation of every position in the input to create these three matrices. They're called Q, K, and V for query, key, and value. And let's consider a single query, Qi, which corresponds to a single desired output that we want to relate to all of the inputs. Now, if we multiply that Qi by transposition of K, we get a 1 by L vector, which corresponds to the weights of each of the inputs with respect to that output. And if we normalize that vector so that it sums to 1 and multiply it by the V matrix, then we can get a weighted sum of all of these representations for each of the inputs. Then we can expand this now to consider the full Q and get the sum for every input for every desired output, and this gives us an L by L matrix of weights, which is a directly calculated influence between inputs and outputs across the sequence. So in a series of easily parallelizable matrix operations, and in a single step, we can directly compute relationships all the way across the sequence. Yeah. So this
maybe a little more intuition why these are called query, key, and value.
Sure. So the query is a vector that represents the desired output, and we match that against each of the keys, which are the inputs. So we're just like searching across those keys and trying to see which ones match most directly with that query. And then so we can get a vector of weights for that for each of the inputs for the keys, and then use those to weight the value vectors. Yeah. Yeah, so the concept of attention was first used in combination with recurrent networks and convolutional networks, but an innovation in 2017 was to do away with the recurrence and the convolution and just use an attention-only network, which is called transformer. And it turns out that this has some advantages in that the model can now look at an unlimited context and look at positions that are unlimited distance away and do this all in parallel across the sequence, and it takes only one computational step to relate to positions. And kind of the only major disadvantage of this approach is that we now have this L by L matrix of weights, and the computation required to do that is L squared now. Nevertheless, it's nice because we have a fixed number of parameters that we can use to do this L squared computation. So this long-term distance dependence means that the model can potentially generate a coherent text that has a consistent message all the way across a piece of text. And this was applied very, very recently in the GP2 model from OpenAI, where they trained a model on a very large set of web text articles, a very diverse set of text, and it used a very large model, and then now they can generate new text. So this is where they prompt a model with this prompt about a scientist discovering unicorns that are in this valley in the Andes, and discovering that these unicorns can speak perfect English. So given this prompt, the model first says that, okay, this population of unicorns has a name, Ovid unicorn, and okay, they were previously unknown to science. Then this evolutionary biologist from the University of La Paz, which is not information that's in the prompt, it's kind of come up with this in relation to that prompt. They were exploring the Andes and they found this valley, and there's this long article, and it concludes by saying that Perez is speculating about how we could know that the aliens are, or if the unicorns are descendants of some alien race by sequencing their DNA. And then he speculates about the implications of unicorns that can speak English. So what this shows is that this model can generate very coherent articles using these long-term dependencies, and it has knowledge of related concepts beyond just sentence structure, and it can also incorporate
what happened with the other 9 tries? It says 10 tries.
Yeah, it is. This is a selected output out of 10 tries. I don't know what those other ones look like. I'd suppose that they're probably just a little less funny. Yeah, so this is a powerful enough model that OpenAI has decided not to release their full model that has about 1 billion parameters on the order of. But you can check out some other examples of generated text on the website. So in summary, I've gone over a set of 3 state-of-the-art or current approaches to modeling sequential data. There's LSTMs, dilated convolutional networks, and transformers. And there's a set of trade-offs with regards to the receptive field that you want, whether it's finite or unbounded, whether you want very efficient training. I assume that you would, but sometimes if you want an LSTM, that's a consideration as well. Whether the model is able to aggregate dependencies in a limited number of computational steps, and also whether there are hidden representations that for each output that are a fixed size or a variable size. So now that I've gone over how we can model P of X, now how do we sample from P of X, or how do we generate new examples? So we want to generate high probability examples from P of X that are kind of representative of what you might see if you got examples from the generative process. Now, the maximum of P of X is intractable, because usually this would constitute enumerating over all possible x's for any reasonable set of inputs, or any natural set of inputs in language. There are a lot of variables here, so that's intractable. And also, the maximum of the distribution may not be representative. This is just going to high-dimensional statistics and comparing the mode of a distribution with samples from that distribution. So methods that can be used to do this fall into kind of 2 broad categories. One is greedy generation, where we choose highest probability choices at steps along the sequence, and the other is just sampling. So greedy search is where we take the argmax at each position. So in the series of conditional probabilities, we take the highest probability x and use that to generate the next. This is not an exact method. It doesn't find the highest probability, and it doesn't tend to, it won't find very diverse examples. And also, it's biased towards common words or common letters. So a way of increasing the diversity is to do beam search, where instead of choosing the single top choices, you could propagate multiple beams where you're choosing the top k choices and or expanding each of the beams, each of the choices for each of the beams, and then choosing the beams that have the highest probability as you go along the sequence. So this is an approximate decoding strategy, but there are ways of increasing the diversity. So because we're choosing the highest probability choices at every step, regular beam search tends to converge on a single path, but there's strategies such as diverse beam search that increase the diversity by adding a penalty for similarity between beams. So this is good if you want to generate a few representative examples that are sampled from P of X. But if you want to generate a large number of samples, probably a better way is just to directly sample from the model. So this is called ancestral sampling or forward sampling, where at every step along the sequence, we sample from the conditional distribution and use that X in downstream samples. So this is an exact method of sampling from P of X. And even though it has no guarantee of optimality, and there's also the issue that noise can propagate forward. So if a very unlikely X is chosen at one of the steps, then this might sort of confuse the model or lead us to generate unlikely X's at later times, resulting in a low probability X sample. So what we can do to address this is to tune the model with the temperature. So this is where the categorical distribution at every step is exponentiated. So each of the probabilities is exponentiated by an inverse temperature, and then it's normalized. So if the temperature is decreased to 0, then this approaches the argmax decoding strategy, as I showed earlier. And if it's increased to infinity, then that would approach a uniform distribution. So there's a natural temperature of 1, which just corresponds to the top equation. And if you want to adjust this, maybe reduce it to 0.5, you'd have to have some metric for choosing the temperature. But nevertheless, this is a way of reducing the diversity if it is too high. Another way of increasing the quality of samples from P of X is to truncate the distribution. This is more common in word-level models where there are tens of thousands of possible words to choose from, and then maybe we truncate the distribution to the top 40. It's not as common in character-level models where there's maybe 20 or 26 characters, or give or take. Okay, so now we have a way of sampling from the model. What do we do if we want to move this model into a biological context? So what works in natural language processing might not work in biology. In general, natural language processing models are in the underfitting regime of this graph here, which is when you increase the capacity of the model, the error reduces or decreases up to a certain extent. And so the training error will tend to decrease continuously as you increase the capacity of a model by adding parameters or increasing training time, et cetera. But the generalization error, or the error on a separate held-out test set, will increase after a certain amount. With a very large model, this would be called overfitting. So in natural language processing, there are a lot of examples for every bit of data. So this is in the underfitting regime, and most of the developments are in increasing the capacity of models and finding efficient ways to train them. But biological data can be more limited in most applications. So it's important to consider this when choosing parameters and such. Okay. So yeah, I've gone over the choices if we want to parameterize an autoregressive generative model and ways of sampling this. So kind of the best choices then for a given model would be if you want to capture long distance interactions, you can use a dilated convolutional network that has a big enough size for a given application, or you can use a self-attention network. And when you want to generate a very large number of sequences, you can use ancestral sampling. So I think that, yeah.
So on the sampling side, if there are these long-range dependencies, does that increase the cost of sampling? Is sampling typically so cheap that compared to training, we don't think about it, or are there regimes where this actually will matter?
Right, so are there regimes where sampling is costly enough that, that this is an important consideration? Right, so sampling from an autoregressive model has to be sequential. You need the parents of a given variable to sample from it. So this means that sampling might have the cost of L squared or such, or even greater. I guess it's an application where perhaps it depends on how many sequences you want to generate, but it can be it's something where you could throw as much compute at it, and probably the number of sequences that you want is achievable with that.
And also, I think when we sample a very long sequence, then at some point you ignore basically what we sampled in the beginning, right? It only takes the last part of the samples into account during the sampling.
Right. So if you could truncate the context if you wanted to increase the speed of sampling, then it would be more linear with length. But then that kind of has the downside that it's harder to make a powerful model of the distribution. Yeah, and like the advances in incorporating longer context kind of get thrown out a little bit, assuming that you want something that incorporates them. Okay, yeah.
How often do you notice some kind of modularity as you go in these longer contexts? In other words, do you get to a point where you get to the equivalent of the end of the sentence where suddenly the probability is much more wide open for whatever happens next? And does that help you at all in sampling?
So if we...
I mean, imagine in the limit you took all text, concatenated together. Obviously, at some point you got the end of a bit of Shakespeare in the beginning of a cartoon introduction, and those two things have nothing to do with each other. As if the model is well trained, it will get to that point. It'll basically say, look, anything can come next here. So the probabilities are suddenly wide open. So is that something you can notice and take advantage of to say, okay, if you hit such and such a point, you effectively don't have any long-term behavior. You've allowed for learning a pattern. You've allowed for learning patterns that are longer than anything that actually exists in your data by any reasonable definition.
Yeah, so there is all of the data that the model has been trained on have a limited size, and if you wanted to make a sample that's longer than any of the training data, weird things might happen. Yeah, so there are definitely models that have been chosen such that they can generalize to new lengths of data. So one of these is called music transformer, where so they generated music, and they found that a vanilla transformer, as I presented here, would actually start to decay and would degenerate as the generation expanded beyond the training set size. So what they did was they added these relative position representations to the attention matrix, and this actually like solves that problem. Yeah, so there are ways of addressing that. Okay, yeah, so with that, I'll leave it to Debbie to show how these can be used in biology. Thanks.
Hi, thanks very much for the invite back. And this is actually the first time that I've given this talk, and most of the stuff that I'm going to present, well, you know, the second half is very just fresh out the oven, and we're still exploring. Okay. And of course, because I'm a procrastinator, and I have two titles there, the general one and the specific one. And one of the things I wanted to say before I launch into it, after the lovely primer on a very, wonderful exposition of a huge field, so I was quite impressed actually, that's developing very rapidly, is that one of the things that we try to do that I realize that I encourage you guys to do, and I'm sure you all do already, but really point out that it's sort of interesting when you hear these expositions, you're sort of excited about using whatever the latest model is, right? And applying that to something, some domain that you know already. And one of the things that we've found over the past 4 years since my lab started is it's very much more nuanced than that. And it's about asking, I'm going to say the obvious, but it's kind of true, asking the biological question and then thinking about what the features of it are and what you want to find out and what the models are that you can use or develop to address them. And then you can avoid the hype train as just the hype and really help develop the field for computational biology, which I think we all want to do. Okay, enough of the lecture. So I'm going to talk today about P of X. I'm not going to talk very much about the first two things, but I am going to talk a bit, certainly not very much about 3D structure. I'm going to talk a bit about effects of mutations, fitness landscape, and then I'm going to concentrate on design in the specific area of antibodies. And going to present to you with that some applications of the autoregressive models that Aaron was talking about. Okay. So this is the bit where I do the show-off part, and then I promise you, well, I try to make it over with. Generative models for biological sequences have been very successful for structure prediction. We can fold from sequences sometimes, and sometimes very well, and sometimes not so well. We can even get RNA 3D structures. We can sometimes see order in disorder, where it's very hard to get information from experimental techniques. We can use the same kind of methods to look at not just within proteins and RNAs, but interactions. And we can use these generative models to predict the effects of mutations. And all these are what I would call, these published things by us and many other people, I should emphasize, are using a sort of unsupervised approaches, okay? And so I'm going to start this talk about the design so that I can mention antibodies early by a sort of little intro on the antibodies so that you understand, or it was helping me to understand what the challenges were and to articulate them. Because it was a field I knew not much about before we were challenged with it. So the kind of chronology of us thinking about this in the lab, because although we knew we were making generative models, even with our shallow ones, it was like, okay, well, we can make all these sequences and we can fit them to the distribution, and then, okay, great, we're great. So what are we going to do with that? And then we were approached by somebody who wanted us to think about library design because he wanted these mini antibodies for protein crystallography. And I thought, well, that's a weird specialized thing. Why would you, that doesn't sound like a big, therapeutic area. Anyway, but as I got to understand so we came at it from a very sort of practical standpoint. Can we do this? And I thought, we probably could do that in a week, and then give him the answer. It wasn't like that. So now here's my introduction to antibodies so that we can frame the question we're going to ask that will then lead into the model. So you know all this, that it's a basis of much of modern molecular biology, and also increasingly therapeutics. And this is something I didn't know before I looked it up a couple of days ago, is therapeutics, $230 billion forecast in the next 4 or 5 years. It's kind of huge. But I don't know how many of you in this room are experimental biologists. Hands up. Right, okay, not very many. There has been a lot of rumbling, and more than rumbling, discussion about the problems of antibodies and the overinterpretation of results that comes really naturally from the way in which they're made and has caused a problem in, if you like, in reproducibility for all sorts of reasons. And there's a huge drive to sort of rationalize the search for sort of proteome-wide antibodies, and there are institutes dedicated to it, for instance, like the IPI in Boston. And the idea there is not to have a one-for-one for each molecule, but actually to have many for its conformational states, for its different functions, different cells. And, you sort of think, well, dream on, but nevertheless, that's the declared goal. And they're doing pretty well, although it's early days. So let's just remind ourselves of, because we're going to talk about design, about antibody domain architecture. And we have the heavy chains in the middle, the light chains on the outside. And then we have the smallest unit of that, that's the recognition. There's a functional unit, which is what we do when we make antibodies, and constructing them in the lab for molecular tools or for therapies. Now, it turns out, actually, that these interesting animals have another antibody type as well as those that is only consists of the heavy chain. And over the past few years, I don't know how long it is, actually. Anybody else know in the audience? I think maybe 10 years, it's become increasingly clear that these, on their own, which are now called, instead of calling them mini antibodies or single domain, they're actually called nanobodies, can be used in the same way that we use antibodies. And you can imagine the advantages there of size and perhaps cell penetration and all sorts of other things. Interesting that the phylogenetic distribution of these makes me wonder, I don't know if anybody's ever looked into this, whether or not it's actually wider but hasn't been found. Okay. So, and these are, in the news now, these antibodies, these nanobodies, also for therapies. I think there was something quite recently about a nasal gene spray for flu and for many other sort of genetic diseases and for cancer. Okay. And obviously, I'm going to say this because I'm going to motivate why we need to help them and design it help accelerate this computationally, but they're still hard, including the nano ones. They're hard to make and they're expensive to make. And actually, this is just a quote from Andrew Cruz. He said, 5 years of injecting llamas with this particular antigen he was interested in failed to deliver useful binders. And then here was a picture, actually, of an article in Science of bleeding a shark to get these nanobodies. So, the obvious thing is to use synthetic libraries. And this is what recently has been developed. And synthetic libraries means you don't have to use the llama or the camel or the shark, and allows sampling of, a large number of unique antibody fragments, fact sorting, and theoretically allows a quick turnaround. The important thing here is that it gives you the direct sequence to function relationship that you only get with monoclonals otherwise. So. Okay, so that's just a repeat. And here's the paper that published the, I think it's the only paper that actually published how to do this screen. It was just last year. And so how did they construct the library? You can imagine lots of ways of constructing your starting library. You've got a yeast display library. And what they did was quite manual, but still quite sensible, actually, is to say, okay, let's take all the structures of antibody variables and nanobodies that we have and look at what varies and whether or not we should vary that in the library. And they basically sort of manually altered certain positions and kept other positions still and created a library. And then the library goes round the sort of typical cycle of things in sort of directed evolution or affinity maturation. In this case, you bind to the antigen, you select the binders. Not as simple as this because you often will have to have things that also screen for stickiness, which we'll come to, amplify, enrich, and then go round again. Okay. So the display libraries are great, but have new challenges compared to using the organism, right? As well as some of the older challenges. So a large, you can do 10 to the 9, but a very, very large percentage will be polyreactive and require many rounds of selection against stickiness. Also, poor stability, of course, this overlaps. And it's not clear, actually, from the numbers yet exactly what those numbers are, but it's very, very high. And also, with the first generation of libraries that we've seen, is that they feel there's insufficient diversity, and the validation is still slow. So along come we and say, okay, right, we do machine learning, and we're going to solve this with generative models to design your libraries. Now, if any of you think about this for a minute, when I first heard this, I was thinking about this, thinking, hang on a minute. When you want to design, maybe it's obvious to you, when you want to design something that's going to target maybe a specific thing, right, or many things, how are you going to design a library? Based on what, if you don't already have it? But, so I want you to understand this bit, because it's really important in terms of what we wanted to design here, and this is why I did that sort of long introduction, is that one of the things about these display libraries is the tendency to be polyspecific and not fold. That doesn't happen in an organism, right? In the ones that we sample from organisms. So if we can learn what is the coding implicit in our natural antibodies and nanobodies in those organisms, that we don't know what those rules are, nobody knows. Can we learn that by generating libraries that are diverse from learning from natural things? So what we want to do is generate diverse libraries that have a very low percentage, or relatively low, of polyspecificity, which is their word for promiscuity or, non-specificity, and instability.
Polyreactivity?
I don't know what polyreactivity means. Oh, right. I wrote it by accident. I mean, I think, these words are used very loosely by everybody.
It's too specific. I'm sorry, not non-specific.
Non-specific. So different fields use different words for, like, directed evolution, affinity maturation, polyreactive, polyspecific. I think that the polyspecific is because sometimes you will get, they want it to be more nuanced than just sticky. So, okay? And obviously that can be quantified. Okay, so there's the introduction about antibodies and nanobodies, the motivation for the library generation, which we're also going to talk, I hope I have time for this, about designing models, what kind of models we can use to increase the affinity. So we're not going to leave that out. But I wanted to emphasize this need to really accelerate this field by making these libraries much more efficient. But now I'm going to take a sort of step sideways and talk about the kind of history over the last few years of, what's the prior art for generative models, and can we use the prior art for this problem? So, and of course I did mention this at the beginning, and you all know this, that natural sequences, for instance proteins, can be grouped into families via alignment. where there's some assumption in that alignment about positions of shared ancestry, or at least similarity. And I want to emphasize this because, along with the sentiment I expressed at the beginning, if you can do it simply, do it simply. Just site-wide, shallow generative models have done wonders for biology. Let's not forget. HMMs, they're the basis of the last 20 years of all the domains and all the rest of it. And, that's literally just a product of site factors. And it's the basis of, HMM's PFAM, homology surf motif detection. It can be a generative model itself. And that's now quite old, as you know. Generative models of sequences have also been successful in pairwise models. And this is now getting to interactions. And to cut a long story short, we know that we can model these interactions with a max-ent model, and we can use things like pseudo-likelihood to solve. And this is known as, in different things in different fields, statistical physics, you might call it a Potts model, and in statistics, you might call it an undirected graphical model. And I've come here and talked about those things before. And as I said before, they've been successful for the thing. We don't want to show those off again. So anybody that wants to look at what we and others have done on that, then we should, there's plenty of material on our website and on our GitHub. And now, the question we had was, because people always asked us this when we were presenting these models, where we modeled pair factors just from the sequences and were able to predict effects of mutation or 3D structures. Well, can't you do higher order? What about higher order? Aren't there long-term, other dependencies? So we said, okay, let's think about latent variables. So I'm going to talk about that first. So this is now not going to talk about the antibodies yet. I'm still talking about just generally the generative models that we and others have built. And I want to mention this particular one because I think we talked about it a little bit here before, and we published a paper on it in November. The reason I want to talk about this is because of the way in which we validated the model, which is actually quite important, I realize, given the sort of large numbers of research coming out now, large numbers of papers that are beginning to appear that are modeling, like large numbers of proteins or the protein universe. And if it's not done, one has to think about how one is going to, if you like, validate the model. And in the cases, this one that we're going to talk about is that this one was in an unsupervised latent model, and so we can validate it by using data that we've not used to train at all. Okay, so let's just talk about what we did here, and then I'll tell you about that data, and then we can talk about how we're going to think about modeling the antibodies. Okay, so this was, as I said, a paper we published recently, and John and Adam have now left me, which I'm very upset about, and gone to better places. And the idea here is that you can model something and capture any order of interactions because you can just integrate out the hidden thingy. Really? Can you? No, of course you can't. So you can't just integrate it out. It's intractable because you'd have to account for all possible and for each x. And so walk in the door, and this was not something, this particular, that we thought of as being used. I think the references are on the bottom here that first talked about these. We used a variational autoencoder, because it provides a tractable lower bound on the P of x, okay? And so you have an encoder, which is the approximate posterior, and you have a decoder, which is the generative side. And then you have this thing, it's called the elbow, which is the evidence lower bound. And this is the most general way of putting it, which is now the difference between the probability of x given the hidden and the parameters minus the Coulbet-Leibler divergence of the two distributions. And you can think of it as the reconstruction cost and the information cost. And actually, we developed this somewhat so that we had also prior on the, I'm sorry, I need some water. Is this for me? Okay. The rest is history, and you can read it in the paper, but here we developed this to prevent overfitting and also to give us information. So, but the main thing I wanted to point out here is less about that model, which you can read about, more about how did we validate it. Because now remember now, what are we training on here? We're talking about training on, we were doing this model to say, can we understand the dependencies of this position on other positions? And if we can, what could we do with that? Well, one thing we could do is perhaps be good at predicting the effects of mutations, better than if we look at just single sites or pairwise. So to evaluate the performance of the model, and remember we're training on, this is aligned families of the sequences through evolution, we can compare to saturation mutagenesis experiments. And I purposely put up one here that didn't look too good, so that it doesn't look like we're showing off too much. And that was the prediction, and there was the experiment. Although you can see the tendencies similar. And you can compare this and you can get the prediction, which is the sort of log ratio of the fitness change to the experiment, and this would be the kind of plot that you'd get of all of the points. We didn't just do it with one, we did it with, sorry, I thought that was animated, I apologize, so you can't see what's underneath. Maybe I should take it off. Actually, we're going to visit it again when we talk about the new model. So the point about this was we have a series of mutation experiments that we collected from published material and sometimes have to contact the authors because they don't deposit their data very well. Not always, but sometimes. And we can compare, and these are hugely varied both in the types of proteins and the types of assays, and we can compare this to predictions from the sequences only. And what we discovered here was that it was better than our own state of art, which was published only 8 months before that, which was the pairwise generative model. So that was interesting. It wasn't always better, and some of the things it was not better were actually quite interesting, and that's for another day. And there were things like we'd want to experiment with about, the size, this kind of distribution of the kind of sequences that are input. Okay. But I wanted to mention that data set. And we can also use this as we could with the pairwise model to design. Now back to the main point, or one of the main points, is one of these things, all these models that we've created and others too, and HMMs and motif findings all rely on alignments. And alignments can be, many biological sequences are hard to align, okay? Obvious ones are, so-called disordered proteins, low complexity proteins, repeat sequences, whether actually they're proteins or DNA or RNAs. And all those things actually that aren't the lovely globular proteins that the universe is mostly concentrated on are actually quite hard to align because alignment has a huge number of assumptions in it that have mostly been built off the way we actually known structures and how they work. So wouldn't it be nice to be able to do these generative models without alignments? And of course, antibodies and nanobodies are a good example of not being able to align, right? Because you can align the framework, but why on earth would you be able to align the variable regions if they're all varying for different targets? So it's trying to do something which is not, you could force it, because it's like if you cluster, you'll get clusters. If you force an alignment, you'll get an alignment, but it won't necessarily mean anything and it won't be stable. So this is just the motivation for using the sequences without the alignments. The other thing we have to consider, and this comes to less about the alignment and more about the kind of model that we're going to use, so think of the primer again, and this is something that Aaron mentioned. Biological sequences have long-range dependencies, and this is an example of a protein where the N and the C terminus come together, and here's this is partly to motivate people to start thinking about this. It's a very crude diagram. These models have not been applied across genomes in any way at all yet, to my knowledge. But you can imagine for promoter enhancers, gene regulation, that this could be a method that could, things that don't, are hard to align, or we don't know how to think about aligning them, which there may be some advantage to using natural language processing approaches. So we want to model long-range dependencies without alignment, and we're going to exploit deep neural networks with autoregression. You heard the primer. And the people who've worked on this are mostly Adam, June, and Aaron. And it was Aaron's birthday just a couple of weeks ago, and just coincidentally, he wore a T-shirt that actually matched this toy that we bought him, which he then put on as a necklace. So there we go. Also with Elena, who's here, who was an undergraduate in the lab at the time, John, who you all know, who was part of the beginning of the project, and the Chris Sander, who's also here, I think. Okay. So we've seen this before. We saw it earlier for those of you who came in later. Teasing. We want to be able to model the dependencies as we would with language. And I'm not going to go into that because you've heard it. And so here's an alignment-based model on the left. An autoregressive model would understand what's gone before. And we're going to use, the first thought here was to use, to think about this domain. Think about the sequences that we're modeling and their lengths and how much a receptive field we want them to have, and therefore what kind of model will be best. And the state of art, we thought anyway, because we hadn't read the transformer paper when we started the project, were these, first guess was going to be LSTMs actually, but we used these dilated convolutional neural nets because of this way that you can increase the receptive field by 2 to the n, so where n is the number of layers, okay? And so otherwise you have to have the number of layers for the number of input variables. And so we can generate the sequences using this model, let's say. And how will we know if it's any good? We can use the same trick we used before, which was to use these mutation scans as some kind of ground truth, say the input now is not aligned sequences, but just the raw sequences. And then we can test these autoregressive models on the dilated convolutional neural nets on the mutation effect prediction by, again, the log of the ratio of the probability compared to wild type. So have I done this again? Maybe. If I've done this again and put the main thing out, I'll go in and alter the slides so you can see. So we find that, so the reason we did this was because we really want to design sequences with this, right? Design sequences that are difficult to align, or assess sequences that are difficult to align for effect prediction. But we wanted to use these things as ground truth. We didn't necessarily expect it to do anywhere near as well as the aligned sequences. And it comes out just a tiny bit worse. Have I done it again? Does it? Good. It's gone off this time. So here across the bottom are all the experiments, and here's the Spearman correlation. In blue is the alignment-free model. In black is our variational autoencoder. So you can see the black is doing better sometimes. Okay? Yes?
So for each experiment, there was some readout. Is the protein doing the thing it's supposed to do?
Yeah, and that's a long discussion. Yeah. Because, what we'd like to say is when we don't do very well, the experiment's not very good. Right. Of course. And actually, there's some truth to it, but we tend not to. So what some people do, actually, who've done a few papers on this kind of thing or subsets of it, is if you look in some of the supplementary material, they will have a plot of this biological replica correlation, and it's sometimes like 0.7 Spearman. So then they will adjust what they get according to, which we haven't. So that's one thing, which is the replication of the experiment. The noise. The second thing is, what are you measuring? What if you only measure, and this is a very good example of this was with the BRCA1, the first round of the BRCA1 experiments, where they did 3 experiments. They did one with all the single mutations. They did one where they said, what's the effect of it binding a known protein interaction partner, BARD1? What's the effect on its E3 ligase activity? As a separate experiment. And then a lower throughput version of what's the homologous recombination, which, if you like, is more of the overall function. And of course, we correlated much better with the overall function. And they would say that in the paper themselves, and they actually in the paper said, oh, look, if we put these linear terms in front of each of those, we can get the homologous recombination. But it does matter. Yes, John?
So, this is a really interesting way to measure how meaningful this like unsupervised model is. Isn't there an issue that you need to, like you're trying to, is your sole task to assign probabilities sort of correctly to things that exist, or like put the mask on the things that exist, or is it, because in this task, many of these are meant to have deleterious consequences, and you're kind of hoping that things that are deleterious correspond to things that are less likely, because how you're, in principle, using the generative model, you're just saying, is this not a likely configuration, and then testing to see whether it's a deleterious configuration. But
right.
not all of these assays,
right.
Do you really know that deleteriousness is being measured at the physiological level just because it's measured at the molecular level in the assay? It just seems like there's like a lot
there's a lot going on. Right. Did everybody hear that? He asked me like 3 questions there. Let's do one of them, which is a little bit like the other one, which is, hey, these experiments aren't necessarily in vivo measuring the effect on fitness of this mutation. There may be a ceiling, the dynamic range of the experiment might be wrong, it might be a sigmoidal relationship to the fitness, it might be ceiling, floor, and you see it all the time, and there's all sorts of measurements for doing that. I mean, all sorts of ways one could adjust for that, okay? So we wouldn't expect to do, so we're very pleased with these results, and we know that they're state of art in terms of compared to even supervised learning methods, which is kind of funky. But it's not quite, I mean, even ground truth when we were doing 3D structure, even crystal structures aren't always ground truth, are they? They're only one ground truth in that state. But there's kind of slightly more than these hugely pooled experiments that have, you know, which, if you like, haven't yet been developed by enough labs to have any kind of protocols around them, and agreed protocols. And one of the things, actually, I would say, and we talk about this with our collaborators like Doug Fowler, who does these all the time, and Jesse Bloom, is, well, now we can do these predictions so well, and you've kind of seen this for yourself and looked at it blindly. What about we go to the next stage of what your experiments are doing and actually get metrics for the kind of, use these tools to help design the experiments, but that's another discussion.
For example, going beyond single amino.
Right, and so what part of the universe do you go to? Right, exactly.
Yeah.
And can you predict, flu evolution? Right. So let's get back down to the ground again. And also, so the second part of John's question, I think I heard, or another question that he asked was, well, these are like, okay, benchmarking your models, okay, but even if you did well, does that tell you anything about how good you would be at generating sequences? I think you asked me that too. Well, actually, we don't know. We're starting to answer that, and that's the last bit of the talk, okay? By actually designing them and testing them. Yeah. So one of the other things is just to show you the features of this kind of model is that we managed to get off people who didn't publish it. I think we had to write to these guys to get this data. Am I right, Aaron? Do you remember? Anyway, sometimes if they've got sort of deletions and things like that in, they don't publish it with the data. So here and our models, our alignment models don't automatically, I mean, you could model it, but it would do very badly. Here we show that obviously, if there's no alignments, it can model what the sequence is that they put in. And here's a reasonable correlation with their experiments for deletions, which, by the way, weren't all deleterious. Okay, so back to designing antibodies using alignment-free generative models, and I want to especially thank Ashish and especially Andrew Cruiser here, who's been an invaluable collaborator on this and work very closely with he's doing all the experiments, not very closely with us on the experiments, all the experiments. We're also now working with the Institute of Protein Innovation in Boston, whose goal is to do make antibodies, many diverse, specific antibodies to states of exposed proteins. I mentioned before that why do we want to do these libraries? Because the libraries are dominated by stickiness and by unfoldedness. And so one thing we thought, one way to test it is to use this model that we have, train on some natural sequences from some llamas, for instance. In this case, we're doing the mini antibodies. And can we generate a much, much smaller library and still get the same results? And this, you know, we went for 10 to the 5, went for like 180,000 we ordered, didn't we? Versus their 10 to the 9. It's quite a tall order. And so this is our first try. And I'm just going to tell you just the sort of topology, the architecture of the model here. Because I don't think we covered that in the primer particularly, did we? No, I mean, it's just an extension of it, but it's, you know, you would input the sequences. So we're going to train on natural. Did I actually have the training? Maybe my training data is next, but here's the architecture. We're going to input sequences, and we're going to have then a convolutional block, which is a dilated convolution with 9 layers. So the receptive field of one block turns into 512 residues. Now, you can start to see how we were thinking about this in terms of the length of proteins, and we wanted to increase this, and so we had 6 convolutional blocks, and gave us a total receptive field of, I think that's right, is it? 3,867? Yeah. I think that's right. And I apologize for this because there's something actually a little bit wrong with this slide, so I do want to do full declaration. So at the top of the out, I've got a sequence, but actually what you're going to output, of course, is a probability of things in that position, right? So like if you can imagine a logo there rather than that. Okay, I mean, obviously this, I could have got away with it, you wouldn't have noticed and said, oh, this is just one sequence that I'm outputting. But in training, that's what it's going to be doing, is making a sort of logo. But the logos are multidimensional because they're going to depend on what went before. Right, so we train on 1.2 million natural nanobody libraries, which we got from this paper in a database. And let's talk about what we wanted to do here. So what we thought was instead of just try, we thought we'll make a library that's 2 or 3 or 4 orders of magnitude less than they normally make. And we could just generate those. But we thought, well, we're worried about the diversity. We want to optimize the diversity. So, I mean, we could now increase this, but this first round, we generated millions of variants. Or, I don't know, was it, did we do it 10 to the 8? Did we do 10 to the 8, 10 to the 7? Can't remember the number, but it wasn't 10 to the 9th. This is just not for the library. This is just, it will be for the library, but this is just what we're in silico generating. And we're going to just do it conditioned on the rest of the sequence, but now just for CDR3, which is the variable bit. And this is sort of not relevant here, but then we can remove sequences that have got reactive motifs. And then what we want to do, and this is part of the, is select the most diverse members from that library that we've generated. And, oh, shoot, can't do Hamming, haven't got an alignment. So what we're going to do, we're going to use a thing called birch clustering. I'm sorry, I've got to get some more water. Which is like a Kamer thing. So you've got 8,000 threemers, and we did threemers, and you've got, so it's about, what is that, 8,420 Kamers. And then each one of these is a vector. Each one of these sequences is a vector of these Kamers, and then you can use, I'll tell you in a minute what you can use. Here we're just talking about ideally, and it's a bit of a baby picture. So ideally, you want it to be a little bit different from the naive library. You don't want it to overlap completely because then it's going to be too similar, and then what have you generated? The same library. And you don't want it to be so far away that, have I got the, yes, I have got that, okay. Well, I haven't got much longer. I better get to the point, haven't I? Nearly there, promise. Right, so we said, okay, we use this form of clustering, and then we can use the cosine distance between these vectors to select the most diverse, right, or representatives in a number of ways. And we can look at the features of sampling in a different way. And here, just to show you, here's the cosine distance between the natural library of the camel. I don't know if it was camels or llamas, this one. Was it a camel repertoire? Yeah, a camel. And then you can see here that this is average, so, okay, that our distance of our chosen ones is bigger, okay, on average. So each one has got a bigger cosine distance to the average of all others. No, each one compared to all others, the average of it is bigger than the natural library. And then that's the distance. The orange plot is the distance of one library to the other, okay? We also want to look at the minimum distance, so, because we want to see if we're just like overlapping, and we're not. And it's quite interesting, this actually, because I didn't expect quite such diversity. And there we are, we ordered it. It's the first time I've ever ordered sequences, actually, so I'm quite pleased with myself, really. It's been, I think that's about 3 months ago, it's been tried. Here's results straight out the oven, and that's why it's not animated, and it looks as though it's straight out of our plot or something. So this is just to show you that, in fact, what the expression level, that's the first thing we're going to check. And Connor, who's a postdoc in Andrew's lab, did these experiments and found that and this is our first try, that the 40% of ours compared to 25% of ours of the original one expressed, okay? And actually, it's a kind of, the blow up on the right side of the pairs is just to show you this is a cleaner cutoff. So even the lower expressing ones have better expression. And we always expect this peak on the left because, of course, we're in yeast display, right? And just early on, we saw some bindings to serum albumin, but that's continuing. We're also looking at, can we improve binding affinity? As I said, so that was a generative model. We're testing it. We haven't got the final results yet. We have heard that there is some selectivity. So one of the acid tests will be, do we have any that when you put that can discriminate, for instance, between an agonist-bound GPCR and a non-agonist-bound GPCR? You know, that would be like our dream result. We don't know yet, but we are seeing some discrimination. I think that's where we are in the results of that. But obviously, we've gone on, and we're going on to design new libraries and look at the features and think about what features we want to do, and this is iterative. So we know that directed evolution can optimize nanobody binding, and that we can... so now I'm going to talk about... I don't think I've got time for this, really. What I wanted to mention is, it's kind of a bit cheeky, really, is to talk about... we can think about now, how can we improve the affinity? Can we accelerate finding the right bind? I mean, if we could, even doing the first thing would accelerate this process hugely, because the yeast library, the size of a football, is very different from a yeast library this size, and it means you can do many, many, many more experiments. But wouldn't it be nice if we could think of a model? So here we're thinking about combining these approaches to do deep sequencing on these libraries and to fit models as we go along, because sequencing is cheap. And we just looked at this as a very simple, it doesn't look so simple the way it's written here, but it is actually a very simple logistic regression model, and with some sparsity. And the sparsity parameter is based on some work that John Ingraham did, actually, with another project in mind a couple of years ago that was published, I think, in ICML. And that's the reparameterization trick for that, which you can read about. The main thing to tell you here is that the model predicts the increasing selection pressure, and in this post hoc, emphasized post hoc, so this is supervised, This is learned from the data, unlike all the other bits, if you like. We did find some, we found that a model like this can recapitulate, which is what we would expect it to do, the increased affinity, and in particular orders with particular structures, which was nice. And we're going to be developing that, obviously. What we'd like to do is think about the integration of the models, and that's something to talk about. The model recovered previously identified advantageous mutations. They were right near the top of the ones we found. Okay. Okay, I talked about those things. I talked about that. I'm going to just do this, and then we're going to stop. This is to inspire you all to work on other things with these kind of models and think about which ones of them might be appropriate for which part. But there's all sorts of areas of design, not just antibodies, that we can think of. And lots of very important large proteins that are involved in things like, differentiation and transcription factors have got these large disordered regions on them we might think about, we want to model and design. And then there's this whole field of membraneless compartments, which is a relief to cell biology that it has this new field to go on about, actually, because otherwise it's been invigorated by it. And it's actually very interesting. So, and it's often involving these RNAs and also proteins with these disordered regions. And these are ways to model. Deimmunization of therapeutics in design, and something I haven't talked about today, but I'm very involved with a fantastic new project on trying to design biostasis-inducing proteins. Gene regulation. We've hammered it with everything over the years. It's the thing of I don't know how many conferences, and why don't we try some of these models on with the right questions for this question, for that area. Genome-wide epistasis. So far, we've looked at that in bacterial sequences, and we've used just the pairwise model to get coupled sites in order to increase the power to be able to look at epistasis in relation to things like antimicrobial resistance. And we want to develop that with these models that don't rely so much on alignment, especially for, if imagine you have accessory genomes that you want to look at that don't necessarily align well. And I've got to mention neurodegeneration because it's actually a field that we're developing strongly in the lab and it's close to my heart. And we're, I think that this is an area, so multi-scale thoughts to do with these models. I want to thank everybody in my lab hugely. I've got to get rid of that picture. It's like from the first year of the lab. Nobody's in the lab anymore in that picture except me. And some of the people in behind are, though. And code is available for everything I mentioned. Oh, I don't think we've put the other thingy up yet, have we? Because, as I said, it's fresh out the oven. The evcouplings.org site has the undirected graphical model for mutation effects and 3D structures. It's done together with the Sander Lab. And I want to thank my collaborators, Andrew and Ashish, very much for getting me interested in the antibody problem. IPI for starting to collaborate with us on a very exciting protocols that they want to accelerate. Christiana again for the other stuff as well. And Pam Silver for her incredible collaborations to do with synthetic biology. Okay, and I'll leave that on and I'll finish. Oh, did it. I think that's the first time ever I've got through my slides.
5 minutes early, actually.
All right.
Is there anyone who wants to ask a question, perhaps about one of these areas?
Or anything, or come up to me afterwards, or write to us. Or...
We have a minute or two.
Yeah. Any questions? Yes.
So if you think big and think about the computational, I mean, even bigger, right? Because this is amazing. And think about designing, say, an entire genome. What kind of computational requirements would that take, and what kind of input data would that take?
Well,
I mean, like, I want a mammal.
Right. So okay. Right. So how many balls of wool does it take to reach the moon? 1 if it's a big one. I don't know how to answer that. I mean, it's like, okay, so, but it's, so there are bounds that one could put on that. I suppose, and we haven't done it. I don't think that actually generating somewhat diverse bacterial genomes would be that difficult. I'm not going mammals yet.
In the epistasis point, are you imagining that these generative models could give you a sort of prior on just
what to test in
to
the
reduce your hypothesis space, or give you a leg up into what to test?
So it's the oldie game. Yeah, it's the oldie game which people have tried to play in other ways with human GWAS and all sorts of things by grouping things in pathways or locations to some very limited success. We want to do it systematically, automatically, and we want to actually integrate the model that we do the space reduction with the association, which at the moment we have as a separate piece.
And perhaps for one protein at a time, or are you thinking bigger in that example? Epistasis.
You mean genome-wide epistasis?
Oh, you want, yeah, sorry.
So we did that with whole genomes, bacterial genomes.
Yeah, that's the gonorrhea.
Yes, which wasn't actually the whole, as you know, it was only the columns where the allele frequency was more than X. So it's a total cheat for me to say that, because say it was, I don't remember, what, 100,000 columns, whatever. So it's a very small genome. And imagine you took, the UK Biobank and we did all humans, what would we be looking at, right?
In the beginning of the talk, you mentioned that you want to learn like the code of antibody sequences, for example, and this also implies learning maybe more like explicit rules, kind of like if you have 5 lysines and some space in between and some other thing, then it will bind to this protein or so. But these kind of like more black box generative models, we can like generate many super nice sequences with nice features, but it doesn't have explicitly about like the design process of the generative process of these sequences.
Are you replaced in the audience? So you're singing my tune here. So did everybody hear what he asked? He said, okay, really, we kind of wanted, you talked about what these dependencies might be that nobody's looked at between what's allowed to be varied and the framework, if you like, over, very different evolutionary time spans, or in the body, 1 over, whatever, much longer. But you can't open the box. Did everybody have that in the US? When I was a kid, there was this thing called open the box. Anyway, we can open the box, right? Because we can look to see, we can do all sorts of analysis, which we've just started to do, to look at what is happening about those dependencies. Now, when we did the variational autoencoder work, that's actually one of the things that we designed into the structure of the model, was reporting, if you like, was learning things that we wanted to understand about the dictionary of what was allowed to interact, or the positions that might be grouped together. And we haven't thought about getting the model to do that, but we've thought about analyzing the sequences that come out to that effect. Because now we've got such a large number of diverse sequences that we can make, and we'll have some idea of the validation of those soon, we hope. Yes, John?
I have no idea what the last bullet has to do with this, so I want you to
I know,
tell me.
I just put it on, I thought somebody will ask me. Okay, so I put single cell to tissue because I think that's what we should all be doing. And the neurodegeneration isn't really a subset of that, but if I do it in any area, I want to think about it in that. But really, so this is a long shot, okay? Everybody knows about somatic mosaicism. So this is not about the RNA and pseudotime and multidimensional scaling, okay? Because I know you know all of that better than me, nor is it about the topology of any of that, because I know excellent people are working on that. This is about rather more uncharted biology, right? So if you think about somatic mosaicism, we've probably hardly touched it. And we know that it occurs in the brain. And I think the extent of it has been very hard to see because single cell sequencing at the DNA level is actually very hard. Somebody called Chris Walsh in Boston is an expert on that, and it's sparse, and there are problems, and blah. But, as those techniques improve and we begin to see, so, we know what happens in cancer. Right? A lot about the evolution of clones, right? What happens in the body? And so I want to think about the evolution of sequences. How am I going to do that without the information? Oh, yeah, all right. No, it's just my excuse to put it on the end. I'm going to do it, whether I use generative models or not. I like generative models because they have to make something that can be tested, or they have to make something you can look at with another thing. I mean, you know, I find it's getting at the black box thing a little bit. There's ways of unwrapping them to think about if you want some mechanistic understanding or fitness landscape understanding that then allows you to design other things nearby, rather than more obvious things that we can do with one of these wonderful machine learning methods, which lots of people are doing, and I do less of it, which is the classification problems. Okay, thank you.